Hello friends, welcome back to in my YouTube channel that is BiologyCenter. Today I am going to teach you enzymes. Before starting this topic, first of all you have to know that enzyme is also called biological catalyst. Okay, so it may be positive, it may be negative. Okay, but when you're talking about a positive catalyst, then it will increase the rate of a particular reaction. Okay, but when you're talking about a negative catalyst, then it will decrease the rate of a particular reaction. Okay, so let's see the enzymes. Okay, almost, please do remember, almost all enzyme is made up of protein. Please do remember, made up of protein. Okay, almost all enzyme is made up of protein. Please do remember, I'm not talking about not all. Okay, not all, but I'm talking about almost all enzyme is made up of protein. So that's why there is exception. That is, except ribosome, that is made up of RNA. Okay, now let's see. We're talking about an enzyme. The enzyme is made up of protein. Okay, so the protein has different form of structure. Okay, that is primary structure, secondary, tertiary, and quaternary. Okay, so let's see the primary structure of protein. Primary structure of protein. Okay, as I have stated earlier, primary structure is the linear chain of amino acid. Okay, when the many amino acid polymerizes to form polypeptide. Okay, which contain N terminal to C terminal. Okay, now let's see. This is the primary structure of protein. Okay, now let's see the secondary structure of protein. Okay, it has two forms. Okay, when the primary structure coiled upon itself and form a helix-like structure. Please do remember, when form a helix-like structure, that is called alpha helix. Okay, but when you're talking about a two primary structure are parallel to each other. Okay, running parallel to each other, in which hydrogen bond are formed. Okay. Now these hydrogen bond stabilize this structure, that is called beta plated sheets. Beta plated sheets. Okay. Now let's see the tertiary structure of protein. Tertiary structure of protein. Okay. Now when a primary structure fold upon itself and form a hazard-like structure. Please do remember. Let's see here. That is N terminal to C, which appears like a hollow woolen ball-like structure. Hollow woolen ball-like structure. Please do remember. Okay. Due to which, talking about tertiary structure, then it shows very high extensive folding. Okay, that is extensive folding, due to which crevices, pocket, and loops are created. Let's see here. All these things are, that is, crevices or pocket or loops are created. Okay, which provide the activeness of an enzyme. Okay, provide the activeness of enzyme. Okay, and these activeness are the substrate binding site. Okay, substrate binding site. Okay, now that's why tertiary structure of protein is called most biologically active form of active form of protein. Okay, now let's proceed further. Okay, now let's see the difference between inorganic catalyst and organic catalyst. Organic catalyst is called enzymes. Okay, please do remember. This is enzyme. Okay, when talking about an inorganic catalyst, then please do remember it works very efficiently in very high temperature and pressure. But when talking about an organic catalyst, okay, then it works very efficiently. It works very efficiently in normal body temperature. Okay, I'm talking about a human. Okay, that is 37 degrees Celsius. Okay, but when we increase more than 40 degrees Celsius, then the enzymes will denature. Please do remember, enzymes will denature. Okay, now let's see. But some thermophilic organism, please do remember, some thermophilic organism which can resist, okay, more than 80 to 90 degrees Celsius easily. For example, as the name indicate, okay, or the name itself, thermo means heat and philic means loving. Okay, let's see the example. That is thermoacidophile. Thermoacidophile. Okay, it is a kind of archaebacteria. Archaebacteria. Okay, now let's proceed further. Okay, different kind of chemical reaction occur in an enzyme. Okay, so let's see the chemical reaction. Chemical reaction. Okay, basically two different type of changes are occur in a chemical reaction. Let's see the changes. Okay, one is called physical changes and the second one is called chemical changes. Physical changes and chemical changes. Okay, physical changes are occur without breaking the bond. Please do remember, it occurs without breaking the bond. But when you're talking about chemical changes, then it works with breaking a bond. Okay, in which all bonds are break and new bonds are formed. Please do remember. But when talking about physical changes without the breaking bond, okay, let's see. That is change in their state. Change in their state. Sorry, change in their shape. Okay, for example, talking about a reaction is linear, then it becomes curved. Okay, this is the change in their shape. Okay, the second one is that is change in their state. change in their state. Okay, for example, let's see here, solid become liquid. Okay, and the next one is liquid becomes gas, and the vice versa. This is the change in their state. Okay, now but talking about chemical changes, then all bonds are formed, sorry, old bonds are break and new bonds are formed. Okay, for example, let's see, talking about barium hydroxide, that is BaOH whole 2 plus H2SO4, it will form barium sulfate, that is BaSO4 plus 2 molecules of water. Okay, now all the bonds, that is barium hydroxide, that is all bonds are break and new bonds are formed between barium sulfate. Okay, in the same fashion, talking about starch hydrolysis. Starch hydrolysis takes place in the presence of water and it forms glucose. Okay, now this is the reaction, that is chemical reaction, which is old bonds are break and new bonds are formed. Okay, now let's proceed further. How you will determine the rate of a particular reaction? Either it is physical or chemical, whatever. How? Let's see. Determination of rate of reaction. How you will determine? Okay, now let's see. First of all, we are talking about a determination, then let's see. This is a reactant and this is a product. Okay, reactant will form product. Okay, let's see. With respect to product, okay, I'm talking about with respect to product, then rate can be says that rate is called, that is rate of formation of product per unit time. Okay, this is the rate with respect to product. Okay, when you simplify this, then it becomes, that is delta P by delta T, and it will always positive. This is the formula. Okay, it will always positive. Why? With respect to time, product will increase. Okay, now but with respect to reactant, we can say that rate equal to the rate of consumption of reactant, that is per unit time. Okay, but when you simplify this, it becomes delta R by delta T, but it will always negative sign are present. Why? Because with respect to time, reactant will decrease always. Okay, now let's see the graph. Okay, y-axis represent reactant or product. Okay, and x-axis represent, that is delta T, time. Okay, as I was stated earlier, with respect to product, it will always increase. Okay, but when talking about reactant, then it will always decrease. Okay, but one time will come, then reactant, that the rate of forward equal to, this is the reason, rate of forward equal to rate of backward. Okay, this is the graph of reactant and this is the graph of product. Okay, now please do remember this graph is very important for chemistry point of view. Okay, now let's proceed further. Rate is also called velocity. Okay, please do remember. Rate is also called velocity. rate is also called velocity. Why? Because if a, if the direction is specified. Okay, for example, we're talking about a speed. Let's see here. A speed is a scalar quantity. Okay, but when talking about a velocity, then it is a vector quantity. Vector quantity. Okay, so it has a specific direction. Okay, now let's see that. When talking about the rate of a particular reaction, then many other things which influence the rate of the reaction. Okay, but when talking about temperature, okay, when we increase 10 degrees Celsius, okay, when we increase 10 degrees Celsius, then rate becomes double. Rate becomes double. Please do remember, when we increase 10 degrees Celsius, then the rate becomes double. For example, let's see, at 20 degrees Celsius, if rate is 1, this is the standard, okay, at 20 degrees Celsius, if we consider the rate is 1, then when we increase 10 degrees Celsius, okay, that is at 30 degrees Celsius, the rate becomes double. Okay, in the same fashion, when we decrease 10 degrees Celsius, okay, it means that 10 degrees Celsius, okay, the rate become half. Okay, now let's proceed further. Some reaction are catalyzed and some reactions are uncatalyzed. But when talking about catalyzed reaction, catalyzed, that is catalyst reaction, catalyzed, catalyzed reaction is very much faster than uncatalyzed reaction. Uncatalyzed reaction. Okay. Catalyst and uncatalyzed reaction. Okay, let's see. We're talking about CO2 plus water molecules. It form a H2CO3. H2CO3. This is called carbonic acid. Okay, that is in the presence of carbonic anhydrase. Okay, that this reaction is occur in cytoplasm. Please remember, cytoplasm. Okay, now this is the reaction. Okay, so without the carbonic anhydrase, that is without the catalyst, please remember, 200 molecules of H2CO3, that is carbonic acid is formed. Okay, which take 1 hour. Okay, 1 hour. But with the presence of carbonic anhydrase, please do remember, the presence of carbonic anhydrase, that is 6 lakh molecule of H2CO3 is formed in 1 second. Please do remember, in 1 second 6 lakh molecules of H2CO3 are formed. Okay, so it dramatically increased, that is 10 million times. Please do remember, 10 million times faster. Okay, so the power of enzyme is incredible indeed. Without enzyme, it cannot sustain a life. Okay, of oxygen, glucose is converted into 2 molecules of pyruvic acid. Okay, but we're talking about in absence of oxygen. In absence of oxygen, glucose is converted into lactic acid. Lactic acid in a skeletal muscle. Okay, But in the same thing, that is 10-step process, we're talking about in yeast. In yeast, it form a ethanol or alcohol by the process called fermentation. Okay, so the main point is that in a different condition, different product are formed. Please remember, in different condition, different product are formed. Okay, now this is how this topic in enzyme topic is completed. Thank you guys for watching my video. If you do really like this video, then please subscribe the channel and share with your friends.